Thanks a lot for the introduction and also for the invitation to this really fantastic meeting. Okay, so I'll tell you today about how we've been thinking about spatiotemporal design principles in multicellular systems, such as tissues, and how to basically decode some of these principles and organization patterns back from biological data. So when I'm talking about design principles for what? So we've been thinking a lot about collective multicellular behavior that has to do, for example, with self-organization and what are the underlying rules for cells and cell-to-cell interactions that lead to these global stable configurations. We've been thinking about collective computation in tissues, how cells compute different outputs to an array of changing environmental inputs, and also how spatial and spatiotemporal pattern formation form over tissues, for example, in terms of gene expression, and what are, again, the underlying rules and mechanisms that can generate different types of patterns. So we're basically, in our group, we're going both the forward and the inverse directions, thinking about these problems, starting from basic models describing self-organization and pattern formation, and trying to identify signatures for these behaviors in biological data, and also the reverse way, which is what I'm mostly going to focus on today, how to start from biological data and infer back what the cells had computed, what type of interactions were there. And in order to do that, for basically any of the questions that I mentioned here, computation and self-organization and information processing in multicellular systems, we need to know how the cells were organized, both in space and in time. So what I'm mostly going to focus on today is how to go from biological data, such as single-cell RNA sequencing or spatially informed transcriptomic data, to infer how cells were organized in space and time in the original tissue. So this is difficult, and as probably many of you know, there are gaps in the type of things that we can measure, or that experimental biologists can measure, and the type of questions we're interested in answering do not completely overlap. So for example, in single-cell RNA sequencing data, to measure the cells need to be dissociated from their tissue of origin. We can only measure a cell once, and therefore we lose both the spatial and temporal context of the cells, which in general, to infer models of collective behavior, these are the aspects that we're mostly interested in. So one of the things that we're trying to do is to close this gap computationally. And we showed that we can basically go from this type of representation of single-cell RNA sequencing data, where we have information about the gene expression levels or activity levels of different genes for many different cells, go back to understanding or inferring how the original tissue was structured, how different temporal trajectories, which temporal trajectories the cells went through, such as the cell cycle, circadian rhythm, and for cell-to-cell interactions and gene interactions. So as an example, which is very intuitive, I think, I wanted to talk for a few minutes about the specific problem of going from single-cell RNA sequencing data to inferring the original tissue structure, which again is lost in the experimental process because the cells were dissociated. So we've been thinking about a very useful analogy here, which is also just generally fun to think about, which is that we're basically trying to solve a puzzle where we're trying to find the optimal configuration of the different puzzle pieces or cells into their three-dimensional or maybe two-dimensional or one-dimensional, if we have some inherent symmetries in the system, configuration in actual physical space, right? So for puzzle pieces, we basically know that we got the right configuration, both when pieces lock into each other in the right way, and also when we get a picture that makes sense, right? And for natural images, we have very strong cognitive biases that we've developed over evolutionary timescales that help us basically process process natural images and also distinguish between a real image and a transformed image. So intuitively, if I now go out with my camera, take a random picture of anything in this room or outside the room in Montreal, and I show you this image and also 9 different random shuffling of the pixels in the image, it wouldn't be very difficult for you to point directly at the real image. And one of the reasons, for example, is that real images show certain characteristics such as continuity in the colors that we perceive, and also other higher order characteristics. So these are biases, by the way, not only, of course, in our cognitive understanding of natural images, but also in the natural world itself. So we thought about how can we basically transform this problem and biases and understandings to biological systems, basically saying if I now take 10 random or 100 or 1,000 random configurations of the cells that we sequenced, place them one next to each other, say in two-dimensional physical space, can I now distinguish which or rank the different configurations according to which one is more likely to represent the natural tissue? And we basically started with maybe the most basic and the easiest prior that we can think of, which had to do with continuity. In this case, not saying three-dimensional RGB channel space, but in 20,000 or so dimensional gene expression space. Basically trying to find a configuration of cells where cells that are close to each other in the tissue would tend to be more similar to each other in terms of their gene expression than randomly placed cells. This was a hypothesis, and we supported it for a wide variety of tissues where we had information about how cells were placed in the natural tissue. And of course, it's not enough to rank configurations because we can't computationally go over all possible configurations. We also needed a computationally efficient way of doing that, and we did that through an optimization framework, through the mathematical framework of optimal transport, where we aim to optimally transport one distribution, in this case of single cells, into another distribution, in this case of locations. Okay, and that allows us to basically get a probabilistic mapping of cells to locations. Oh, interesting. Okay, we'll just skip that slide. Hope it's the only one that's shuffled. Okay, so that allowed us to now take single cell RNA sequencing data and basically put the puzzle pieces together to reconstruct the original tissue these cells were dissociated from. So this is an example for what you see here are 4 different gene expression patterns over Drosophila or fly embryo. And you see on the top, you can see the experimental measurements for these genes. On the bottom are predictions, and you see that we do a very good job even with complex patterns. And we can do that for more than just the spatial domain and actually generalize this to additional domains such as so spatial structures, but in non-prototypical systems, such as in cancerous ecosystems. In this case, what you see here is a colon, is the colon ecosystem, and then also for temporal trajectories. In this case, for example, inferring the fate of early differentiating hematopoietic cells. Okay. So we can basically take single-cell RNA sequencing data and then infer, say, either the spatial configuration of the cells or their temporal dynamical structure. But that also means that if we do it one step at a time, then it's a little bit similar to this really nice cartoon that I like, where we have 6 blind men and women trying to figure out, or blindfolded, trying to figure out what they're standing next to, right? So each one has a specific angle or a specific projection of the elephant, right? So the one standing next to the tusk thinks it's a spear, and the one on the top thinks it's a fan, and the one on the bottom in the back thinks it's a rope, right? So if we look at our systems from just one perspective at a time, we would get different answers. And one of the things that we realized, which were there all along, of course, is that the cells actually encode multiple layers of information simultaneously. So in the same collection of gene expression of cells, there should be encoded some information about how the cells are organized in space and which types of dynamical trajectories they went through. So some information about the cell cycle and some information about cell-to-cell interactions and so forth. So now the broader question was, can we disentangle or tease apart different layers of information from single-cell data? Okay, so there are different ways of approaching this very broad question, and I'll share with you 2 of the things that we've been working on recently. So one of them has to do with the following setting, which is a direct continuation of what I showed you so far. So say we have single-cell RNA sequencing data, and there's either a very strong signal in the data or that we have very strong priors, and therefore we can extract one layer of information out of that. For example, how the cells were organized in the original tissue, right? But we know, okay, so we have that information, but we know that, as I told you before, the cells also encode simultaneously additional layers of information. So now it becomes something that we've been thinking about as a filtering problem. Basically, we have our single cell data. Can we now filter the layer of information that we already reconstructed or measured experimentally to recover what additional layers of information are encoded by the cells? So a different way of thinking about this problem, as you see here on the screen, on the left, we have, say, the original representation of the data. We can think about a low-dimensional representation, say a PCA space of the single cells, and you see that they're clustered according to one signal in the data, for example, cell type, or for example, their spatial structure, right? So now the question is, can we basically filter that signal out so we can recover the additional signal in the data, in this case, B, or how the cells, so not how the cells are colored, but corresponding to their shapes. And we were thinking about it as basically filtering out the similarities, the types of similarities between the cells that are directly related to the layer of information that we recovered. So if we think about single cell data and we see 2 cells that are similar to each other in their gene expression, we basically don't know whether they're similar to each other because they were located next to each other in the original tissue, or because they're found at a similar stage or phase of the cell cycle, or similar differentiation stage. What is it about them that made them similar? Probably multiple different factors. So what we do here, basically, using a kernel-based projection and filtering method, is to basically filter out the similarity structure within the data, or the covariance structure within the data, that is specifically corresponding to what we already know about the cells. So, for example, filter out the similarity structure of the cells that is associated with their spatial structure, to then recover a new adjusted expression matrix where we can now discover additional signals in the data. So, for example, what can we do with this kind of approach? We can take single-cell RNA sequencing from the liver. In this case, and I'm sorry for the PDF transformation problems. So I'll just tell you what you should be seeing here. So we can take single-cell RNA sequencing data from liver lobules. In that system, there's a very strong spatial zonation signal in the data, and therefore we can actually reconstruct the spatial structure without any additional information or any reference atlas. But we also know that the cells also change their state along the time of day, along the circadian rhythm. So what you see here on the right, on the y-axis, is our ability, basically a proxy for our ability to reconstruct the circadian rhythm based on the single cell information. On the x-axis, you see how many additional genes we need to use, or the size of the reference atlas, or you can think about it as the strength of the additional priors that we use. In blue, you can see how well we do in reconstructing this temporal progression, or the circadian rhythm, from the original single cell data. And in orange, what we get when we first filter out the spatial zonation signal. So here, the spatial and temporal signals interfere with each other, and therefore, when we try to reconstruct the temporal signal, its interference in the original data makes it hard for us, and we do much, much better when we filter out or disentangle the different signals from each other. Okay, so now we're going to take a small detour that somehow has connections to some of the other talks that we've heard in the past few days. And I told you that in this type of approach using SIFT, we can basically filter out data based on our knowledge about a specific layer of information, or maybe we measured it, or in general, we have some annotations for the cells, for example, for how they're organized in space, right? So now the question becomes, can we always trust our annotations, either generated by us or generated by other studies, right? So it's a very common thing, I think many of you would relate to when you're trying to analyze, say, the published data set of single cells, for example, that many times comes with pre-annotations, right? Either annotated by the original study or measured experimentally. And we know that many times there are basically gaps in our knowledge, or gaps in how we basically understand the data. So not all of the annotations reflect the underlying biology of the cells. And we were interested in trying to understand whether we can specifically identify these gaps to then filter the data to only what we're sure about, or basically filter out our gaps in knowledge, and maybe even re-annotate the cells that we suspect. So to make this more concrete, actually, let's go to a different analogy. Taken from machine vision, okay? And we basically No, so we usually, we talked in the past 2 days, we talked a lot about how machine learning and predictive models can be used to enhance biological and biomedical research. And mostly we take advantage of the output, of the direct output of the model, right? In the simple cases, we have a simple machine learning predictive model that, for example, given a cell, predicts its cell type or cell state, okay, for example. Or if we go to these nice images, which are sometimes more intuitive to think about, we have a set of images of dogs and cats, and we have a model that predicts whether we see a dog or a cat in the image, okay? So we decided to use a characteristic of learning models that is usually ignored in this kind of analysis, which is to basically track the training dynamics of the network itself. So not look directly at the prediction, basically take into account that if the model is expressive enough, if we give it enough time, it would eventually memorize the entire data set, but that different types of images would have different statistical characteristics in terms of their training dynamics. Okay, so what does it mean? Say again that we have this very large data set of dogs and cats, okay? So there would be some images in the data set that the model has a very easy way of knowing that this, for example, is a dog, right? So if we look at all of the photos, all of the images that are initially annotated as dogs in the data set, the model would easily predict this as a dog, easily in the sense that over the training process, the probability of predicting this is a dog would basically go up and saturate very quickly and stay up. So it would be of high confidence and low variability in that sense. On the other hand, we would also have images where the model would have a very hard time. So this image was originally annotated as the dog, okay? The model in the initial stage would say, okay, it's not a dog, it's not a dog, it's not a dog. But then if you train it for long enough, eventually it would give up and say, okay, it's a dog, I believe you. Okay, that's basically the overfitting regime in the training process. So we would have, in this case, very low confidence in the initial annotation and also low variability, because for the vast majority of the training process, it would always be predicted as a cat, and then transition into a dog prediction only towards the end of training. And then we would also have these ambiguous or intermediate type of images where the model will not be so sure about. And its predictions would be very variable across the training process. So, of course, we're interested in biological systems, so we can make the analogy here to biological data, and specifically if we're thinking about cells, then if we're looking at a single cell data set, for example, and say that we look at all the cells that were originally annotated as green cells, okay, of a specific type, for example. So this one would be easy to learn. This one would be hard to learn in the sense that I told you about before. And maybe if we measure two of these cells together experimentally, it would be ambiguous. The model would not be sure about it. So that could be either due to technical difficulties or to inherent biological ambiguities in the process. For example, if we have a cell that is transitioning from a green to a purple state, then the model would have a harder time for that as well. Okay, so all of this can be formalized, and this paper is currently in the bioRxiv. We termed our approach annotatability. And we can basically, using this very simple idea, we can basically do a lot, like identifying and also reclassifying misclassified cells in single-cell RNA sequencing data, and also spatial transcriptomics. We can also use it as a basis to reveal continuous trajectories in the data, and also do more like assessing treatment effectiveness and identifying rare populations, such as healthy-like cells in diseased tissues. So here is an example. If we look at single-cell RNA sequencing data of PBMCs, then if we look on the y-axis here on the mean probability of these cells being assigned to their original annotations, the correctly annotated cells would have this kind of easy to learn behavior that I told you about before. The model would predict them very quickly and very robustly. On the other hand, the cells that were misclassified would have the hard to learn dynamics where the model keeps thinking it's a different cell type, but eventually overfits. And then we also have the ambiguous states that are just more variable and slower to learn by the system. Okay, we can also look at the additional cell type configurations, but maybe I'll skip that to say that given this idea, we can take our single cell data, basically follow the training dynamics over the different cells according to predictions of a very simple neural network model to predict their initial input annotations. And then based on their confidence and variability scores that have to do with the training dynamics that I told you about before, we can basically group the cells into easy to learn, hard to learn, and ambiguous cell states. So that allows us, for example, to identify within all the cells that were originally annotated as B cells in this study, we could identify the correctly annotated cells, and here you can see they match their respective marker genes, and also the incorrectly annotated cells, which when we look at a panel of marker genes that we didn't use for this classification, we can see indeed that there is a good basis to say that they were misclassified. And this also works, for example, for spatial transcriptomics data, where we actually expect a much larger fraction of misclassified cells. So here, for example, looking at MERFISH data of the preoptic region, and we look at all the cells that were their input annotation was inhibitory neurons, then we can see the correctly annotated cells that we identified. So they indeed match their marker gene annotations, and then the misclassified cells. And then we can do more. We can actually now reclassify the cells by training, again, only over the cells we identified as correctly annotated, and get this transition matrix from cells we predict that were initially misclassified into each one of the other categories. And here we can see a validation that indeed this transition makes more sense in terms of the marker genes of these cells. Okay, so now let's go back to our main thread and go back to this general question about how can we disentangle different layers of information. So I wanted to tell you about another direction we've been thinking about. So say that we don't necessarily already recovered one layer of information or measured one layer of information that we can filter out. We can actually make use of this very nice characteristic, which is that many of the biological processes that we're interested in are topologically distinct in gene expression space. So we have, for example, hierarchical or tree-like trajectories corresponding to differentiation or developmental processes. We have cyclic processes or cyclic trajectories in gene expression space that correspond to periodic or cyclic processes like the cell cycle or the circadian rhythm. And we also have grid-like structures that correspond to spatial configurations of the cells. So if we have that, then that also means that maybe we can identify signatures that are specific to these different topologies. And then if we identify these different signatures in single cell data, that would allow us to also say what type of biological processes the cells are encoding. So to be more specific than signatures, we specifically looked at the spectrum of the covariance matrix of single-cell data. And for example, if we would just have a random matrix of gene expression where different genes in different cells are not correlated with each other, then we know what we should get in terms of the eigenvalue distribution in that case. It's a basic result in random matrix theory, and we would expect to get the Marchenko-Pastor distribution, which in this case is shown on a log-log plot of eigenvalues versus their rank. And my point here is that this is, of course, a non-biological scenario. If we have correlations within our data, either between genes due to gene-to-gene regulatory interactions, or between cells due to their spatial or temporal configuration, as we talked about before, we would expect different signatures. So, for example, if we have a basic hierarchical model of gene expression, where different cells generate a hierarchy of states, then we can see here that we get a very different signature than what we would get for our null model, our non-correlated null model, and specifically a power law of large eigenvalues. And this is actually something that we find in single-cell RNA sequencing data corresponding to hierarchical processes such as differentiation and development. And we can do more than just identify this in the data and say, okay, these groups of cells encode this type of topology, which is therefore corresponding to this group of biological processes. We can leverage this to also manipulate the data itself. So to filter or enhance different biological processes within our data that correspond to different topological structures. And we did that through a framework that we call SC Prisma, which is basically a spectral template matching optimization framework. And through a series of optimization problems, we basically find the corresponding topologies in the data and then either enhance them or filter that layer of information from our single cell data. Okay, so for example, if we take the same type of single cell RNA sequencing from liver lobels as I showed you before, then this is just a PCA representation of the raw data colored by both different time points along the day wherein the cells were sampled, or colored according to their spatial location across the axis of the liver lobel. We don't use this information. This is just a guide to the eye. It's a proof of concept to show the type of things we can do with this spectral template matching. So in this case, as I told you before, we have two main signals in the data, one corresponding to a temporal trajectory that's cyclic because it corresponds to the circadian rhythm. So if we take all the cells together and just spectrally enhance the cyclic signal in the data, what you can see in the middle column is that we can retrieve a signal that corresponds to the temporal progression of the cell, and then also indirectly filter out the spatial signal that is, which was what you see on the bottom. And then we can also manipulate directly the spatial signal in this data, in this case corresponding to a linear trajectory. And if we directly enhance the linear structure in the data, we can get back a representation that corresponds to the spatial zonation of the cells across the liver lobel. Okay, we can do that for many different types of templates and different types of processes, and use that to manipulate single cell data in general. Okay, so this is, I think, very interesting, these types of approaches that allow us to manipulate layers of information within single cell data, not only because they expose how the cells are organized, say in space or in time or according to other processes, but it also allows us to ask follow-up questions. So for example, if we think about inference of gene regulatory networks, like we've heard in some of the earlier talks today, or inference cell-to-cell interactions, then usually this is done with, there are many different algorithms to do that, but usually these algorithms are applied over the original data, right? And we know that these types of interactions, either between genes or between cells, are context-specific, right? So gene-to-gene interactions, for example, can change along the time of day, or can change according to where the cell is in the physical tissue and the type of, and its corresponding microenvironment and the type of signals it's exposed to. So if we're trying to infer a gene regulatory network out of the whole data, then we're missing something. We're basically inferring a network that never existed for a single cell, and that also makes it a much harder problem. So the point is that if we can now manipulate different layers of information, then, for example, we can take the single cell data, enhance only the temporal trajectory corresponding to, say, the cell cycle, and then infer the gene regulatory interaction structure that is specifically related to the cell cycle, right? And we can do the same thing for the spatial configuration of the cells or any other types of processes. Okay. So the last idea I wanted to share with you is that we can basically generalize what I told you about the fact that different topological structures corresponding to how the cells are organized in gene expression space, that corresponds to different biological systems. We can basically generalize it to infer the biological processes cells are going through in different parts of gene expression space. So I'll do that in a more concrete way through an example. So here we see single cell data for pancreatic development. Basically, we see cells that are first proliferating and then differentiating into their final states. And this is overlaid with RNA velocity information over that data. So our thought was, can we actually predict from this type of information that the cells are first proliferating and then differentiating into their terminal states, not by using any type of marker gene information, which in this case may be known, but in other cases it's not. So not using any information about the genes, but just using the fact that the dynamical behavior of the cells changed throughout this process, and specifically the regime of proliferation. When the cells are going through the cell cycle, we would expect to see a cyclic behavior in gene expression space, and maybe something that's a little similar to a limit cycle in that part, of the space. And then when the cells are proliferating, basically flowing towards their terminal states, we would expect to see something that's more similar to a regime with fixed points, if we think about it from a dynamical systems theory point of view. So to do that, we developed a physics-informed deep learning framework that uses both data augmentations that preserve the topological characteristics of the original data. So here, for example, we train over only data from a simple oscillator, maybe the simplest type of dynamical systems that in this case shows a bifurcation, specifically a supercritical pop bifurcation from a regime where it has a stable fixed point to a regime where it has a stable, the fixed point loses its stability and the limit cycle, stable limit cycle emerges. So if we just take that original data, it's not rich enough to then infer anything about real biological systems or real data in real measurements in general. So we basically augmented the original data from the simple oscillator with topologically preserving augmentation. So the data can look very different from each other, but a limit cycle would be transformed or augmented into a limit cycle and a fixed point to a fixed point. And then we do angle transformation to preserve not the amplitudes, but only the angles of the directions, basically. The single cells are flowing into, in this case, and then have a convolutional neural network with self-attention to predict basically whether, in this case, we would expect that the original system exhibits a fixed point or a limit cycle. We can also generalize this, but this is an example for this specific case. And then if we take the original data, take different cell types, interpolate vectors of cellular flow in gene expression space, then using this framework, we can basically predict where the cells are exhibiting a limit cycle-like behavior or a fixed point. And here is basically our predictions for a limit cycle or proliferating state on the left in red, and a prediction for the differentiation stage on the right. And this is what we get when we use marker genes just for validation, and we get a very good fit between the two. Okay, so the hope, again, is that all of these types of frameworks would help us go back and better understand complex collective behavior in multicellular systems. And I will end by thanking my lab members, and specifically Matt Ritchie, Noah Moriel, Zoe Piran, Jonathan Karin, and Sima Dubnov, whose works I highlighted today, and collaborators and funding, and thank you.
Thanks, Mor, for a wonderful talk. So we have time for questions. So here is one microphone. Yeah. Or you raise your hand, and if you're on this side.
Hi.
Oh, thanks. Wonderful. I had a question about the first part where you mentioned SIFT, and that you regress one signal to identify another signal.
Right.
And if you can dig a bit on the intuition behind that, because for me it was like, if you do a PCA, you just dig in more in components like third and fourth and so on, and you will recover your process B somehow. So how regressing is helping disentangling more than digging into the principal component somehow?
Yeah, so thanks for the question. So this is a really important point to better understand what we're trying to do here. So I'll go back to what I said before about similarity structures between different cells, right? We basically, for the reconstruction works that I talked about in the first part of the talk, we basically use the idea that cells that are similar to each other are expected to be located next to each other, either in space or in time. And of course, there are, it's not always the case for more complex tissues, for more complex programs, in which case we also need to take into account additional priors like reference atlases. But in general, that's the most basic prior that we use. So this was our original view. But then we say, okay, so cells can be similar to each other due to different types of processes, right? Because they're correlated according to different types of processes, either because they're correlated next to each other in space or because they're correlated in time. So the idea here, the idea of projecting out a kernel of similarities is to say, if we have, for example, our single cell data, we were able to reconstruct the spatial configuration of the cells. Now we know how the cells are similar to each other, specifically according to their spatial configuration, right? Because we can actually compute it according to the spatial configuration we reconstructed or measured. So we have the similarity structure between the cells in the original data. We have the similarity structure in the reconstructed data. And now the intuition is basically to filter out to filter one from the other to get a similarity structure that was not basically the gap between the two, right? To capture additional processes in the data. Thanks.
Other questions?
I have a question related to that. So what if your processes are dependent?
Yeah.
Like a cycle only in some part of the differentiation or the zonation
Right.
of the liver. Will you lose that or not?
So this is a great question, and we're thinking a lot about that. So the processes don't have to be completely orthogonal or independent of each other for these types of ideas to work. And for example, I showed you how we used SIFT for the liver data, where we know there are some interactions between the information encoded about the spatial and the temporal signal. So it can work, but at some point, if the two different processes or multiple different processes are highly correlated with each other, we would expect this to perform much worse. And therefore, one of the things that we're currently doing is actually developing methods specifically for this case. So it's not only to show that it works when the processes are correlated, it's first knowing when different processes are correlated or what type of interactions are between different biological processes that cells encode, then to be able to distinguish them or disentangle them from each other. And also better understanding the mechanisms for these types of interactions. So this is a big project that we're currently working on.
Hello, thank you.
Hi.
I have a question about the reannotation approach. How did you ensure that you start with a well-calibrated model where you can trust the probabilities?
Yeah, so the basic idea is to use the simplest, most general model. So specifically, we used a neural network that's with fully connected layers. The default is only 3 layers, very simple, but expressive enough to eventually overfit the type of data sets that we used. And the whole idea there is that all you need to do, and we have different sensitivity and robustness analysis to support what I'm going to say, but all you need is a model that can basically learn the input annotations to the extent that at some point it would memorize the annotations and overfit to the entire data set. So you would have a model that, given a sample from the data set, would predict the input annotation. And we're only talking here about the training phase, right? Not test or validation. So that's the idea.